Good morning, everyone. Today we have a new lesson to tackle. But before that, I would like to introduce our members. I'm Dhainielle.xo, editor, reporter, and researcher.
And I'm the second member, Dhainielle.xo, leader, editor, and reporter.
I'm Cristana Bernabe, assistant leader, scriptwriter, and reporter.
Our first lesson is the cell cycle. But before we tackle this lesson, we will be knowing our objective. Number 1, identify the event that take place in each phase of cell cycle. Number 2, differentiate among the cell cycle points. What is cell cycle? The cell cycle or cell division cycle is the series of events that take place in the cell that causes to divide into the two daughter cells.
Cell cycle has two phases, interphase and mitosis. Interphase has the longest part of the cell cycle. It is the state of preparation. Cell perform basic function. It grows and replicates DNA to prepare for mitosis or cellular division. After the parent cell divide, each of the two daughter cells will pass interphase again. Interphase can be divided into three subspaces, which is G1, S, and G2. G1 is referred as gap or growth 1. It's the longest phase of the cell cycle. During G1, cells mostly grow and the organelles take care of regular cellular business, like synthesizing protein and producing energy. Inside the nucleus, we can find the DNA, organized as chromosomes. During G1, each chromosome is made out of single thin spaghetti of DNA, or what we call chromatid. At the end of G1, there's a cell cycle control point called the G1 checkpoint. It checks if the DNA is not damaged and synthesizing right proteins at the correct amount. If it turned out that there's a reason that cell cannot divide, like having DNA damage, things can go one of the two ways. Either non-dividing stage called G0 phase, where DNA repaired mechanisms try to fix the problem, where the cell self-destruct in the process called apoptosis. If the cell gets through the G1 checkpoint, it enters the S phase. S referred as synthesis. During this phase, DNA is replicating. Each daughter cell receives identical copies of the genetic material. So for each chromosomes from G1, an identical copy created. This happen with the help from the number of protein, both structural proteins and enzymes, as well as energy. Just to be clear, the numbers of chromosomes does not increase. Human somatic cell has 46 chromosomes. Throughout the cell cycle, the amount of DNA has changed during the cycle. So each of the chromosomes will enter at the S phase with a single copy of genetic information or the chromatid. During replication, each chromatid are copied and pasted. So the amount of DNA doubles up. It results to two identical chromatids and to the original genetic template, and they join together at the center, or what we call centromere. But they still make single chromosome. So all the amount genetic information are doubled, they are still 46 chromosomes that contains genetic information. The cell now enter in G2 phase. G2 serves for gaps or growth 2. Even after synthesizing the copies of DNA, the cell has to duplicate organelles so they have enough for both daughter cells. In fact, at the end of G2, the cell will look like a big balloon at the cytoplasm and organelles. Before it can do it though, it should pass the final G2 checkpoint. If there's no DNA damage after replication, it now enter mitosis. Mitosis actually divides the two daughter cells. It involves separating the replicate DNA into distinct nuclei, a process called karyokinesis, and the actual separation of daughter cells to two distinct ways, a process called cytokinesis. Mitosis can be broken down to four subspaces. In order, it's prophase, metaphase, anaphase, telophase, and they are immediately followed by cytokinesis. You can remember this by using please make another two cells.
During prophase, the membrane around the nucleus disintegrates and the chromosome condense and become invisible under a microscope, like 46 little X shape. The condensed sister chromatids make a thin Y and male look like an X. Inside the cytoplasm, two organelles called centrosome migrate to opposite walls of the cell. During metaphase, chromosomes move toward the middle of the cell. It is called metaplate. It is just like an M for metaphase and M for middle. When the chromosome is in place, it is centrosome. Centrosomes send out thread-like projection called spindle fiber that attach to the centromere of each chromosome.
Metaphase, second stage of mitosis, is defined as the stage of mitosis when all the chromosomes are lined up along the center or equator of the cell. Throughout this short 5 to 10 minute stage, the chromosomes are attached to the spindle fibers, and the centromeres that bind the sister chromatids together split apart.
Next, anaphase. The centrosomes start to pulling spindle fibers to pull sister chromatids apart, like a game of tug-war. Since both centrosomes pull with equal force and opposite direction, eventually the sister chromatids separate and reach opposite poles of the cell.
Splitting of the centromeres signals the start of the third stage of mitosis, called anaphase. Anaphase is defined as the stage of mitosis when the sister chromatids separate and move toward opposite poles of the cell. And when this happens, they are no longer called sister chromatids. They are now called daughter chromosomes. This movement happens fairly rapidly, about 5 minutes, with the result that the spindle fibers disappear from view, and a full diploid set of chromosomes is now found at each end of the cell.
Last is the telophase. The nuclear membrane reforms around each new set of 46 single chromatid chromosomes. After that, during cytokinesis, the cell membrane pinch until the two daughter cells separate.
The final stage of mitosis is called telophase. Telophase is defined as the stage of mitosis when the new daughter chromosomes change back into the threads of chromatin and new nuclear membranes begin to form. Also during telophase, new nucleoli appear in each newly forming nucleus. As the final stage of mitosis concludes, the cytoplasm divides in half as cell membranes close up around the two new daughter cells. This final process of cell reproduction is called cytokinesis. Cytokinesis is defined simply as the division of the cytoplasm during mitosis. Between 10 and 15 minutes are required to finish both telophase and cytokinesis. When the entire cell reproductive process is completed, the two new cells are returned to the interphase state, and each possesses the identical genes of the parent cell. These two new half-size cells will then go on to grow larger until each achieves the full size of the original parent cell.
Now, both of the daughter cells can enter the cell cycle once more, if they need to. If they do not, they enter the non-dividing or J0 phase. J0 is a phase outside the cell cycle because the cells are not dividing nor preparing to divide. Example is hepatocytes, which rest in G0 until there's liver damage and go to G1 again so they can divide and replace the damaged tissue. Other examples are neurons spend their entire time in G0. The uncontrolled division of cancer cells cells may develop after several characteristics are acquired, allowing the cell to become neoplastic. 10 hallmarks of cancer have been postulated to describe these changes and are shared by most cancers. We would like to highlight 5 of these hallmarks in this episode. First, cancer cells are capable of sustaining proliferated signaling in the absence of growth factors. In other words, they are able to enter the cell cycle without requiring positive external signals. Second, cancer cells are able to evade growth suppressors. Let's use an example here. Non-tumorous cells grown in a petri dish would proliferate until a single layer is formed that covers the bottom of the dish. A phenomenon known as contact inhibition would then prevent the cells from further proliferation. In contrast, contact inhibition is lost in cancer cells and results in uncontrolled cell growth and proliferation, producing a multi-layer of cells. Another example of growth suppressors are cell cycle checkpoints, whose function is usually impaired in cancer cells and leads to unchecked growth. Third, cancer cells are divided indefinitely. They have limitless replicative potential. This is in contrast to non-cancerous cells of the body, which are incapable of indefinite division. The mechanism behind this lies in the telomeres, which shorten with each cell division. However, in germline cells where repeated division is required, the enzyme telomerase is able to elongate these ends, thereby enabling indefinite cell division. Fourth, cancer cells also show increased genetic instability and therefore have a higher frequency of mutations. This occurs as a result of inefficient repair of DNA damage during the cell cycle and does not induce programmed cell death. Fifth, another hallmark is the disabling of apoptosis. As a consequence, damaged cells are not neutralized. In other words, cancer cells evade programmed cell death. The accusation of these hallmarks and other cells' characteristics are not the result of a single event but the accumulation of DNA damage over time. Let's take a look at the relevant genes. Cancer genes, it is important to note that mutations can occur in all genes. However, in several genes, mutation will lead to tumor formation. These genes can be differentiated into two groups, proto-oncogenes and tumor suppressor genes. Proto-oncogenes are genes whose uncontrolled activation contributes to tumor formation. If the gene is mutated, it is called an oncogene and cell division is facilitated. Oncogenes are, for example, involved in transmitting growth signals such as the receptor for the human epidermal growth factor or HER2 or NEU. In contrast, tumor suppressor genes require the activation to contribute to tumor formation. The most important tumor suppressor genes control the cell cycle and, when functional, inhibit uncontrolled cell division. One example is P53. It has been nicknamed the guardian of the genome due to its importance. Regarding the terminology, you might want to keep in mind that anti-oncogene is sometimes used synonymously with tumor suppressor gene.
There are many types of syndrome. Credichat syndrome. It is a rare genetic disorder caused by a deletion on chromosome 5, characterized by a high-pitched cry, microcephaly, and a flattened nasal bridge. Down syndrome. It is a genetic condition characterized by an extra copy of chromosome 21, resulting in developmental delays and increased health risks. Edwards syndrome. Edwards syndrome, trisomy 18. It is a severe genetic condition caused by an extra copy of chromosome 18, leading to low birth weight, multiple birth defects, and developmental delays. Number 3 is Jacobson syndrome. It is a genetic condition caused by a deletion on the long arm of chromosome 11, 11q terminal deletion, resulting in features such as sparse facial hair, reduced muscle tone, and other physical characteristics. Number 5 is Klinefelter syndrome. Klinefelter syndrome XXY is a genetic condition affecting males, characterized by an extra X chromosome which can lead to reduced fertility and other developmental features. Turner syndrome is a chromosomal condition affecting only females, caused by the absence of one X chromosome, leading to short stature, ovarian failure, and other health issues.